Today I would like to discuss the phenomena of stochastic vesicular release at the presynaptic terminal of neuronal cells. So in part one I will present the basic biology, physiology, and part two I will go into the modeling part. So most of the software, as usual, will be available on our website, Bionumerics, and here at bionumerics.org. So basically, a synapse, as shown here, is made when a neuron connects through the presynaptic terminal here in pink, connect to the postsynaptic one in yellow, and here this is on a structure called a dendritic spine. This is an electron microscopy picture, and you see here, although it's not very clear, region of high density. It's really dark here, which means accumulation, aggregation of many important proteins. And when we look specifically at the presynaptic terminal here, what we see a collection of vesicles that are really to be to go on the surface membrane to release their content, which are neurotransmitter, and through the release of neurotransmitter, they bind specific presynaptic terminal, receptor on the postsynaptic terminal, such as, for example, on an excitatory synapse, on parareceptor and NMDA receptor, which leads to an influx. And we're not going to discuss much here the nature of the influx, which could be sodiums, calcium, ion, such as in NMDA, that has physiological consequences. So today I would like to discuss what happened at the presynaptic terminal for these vesicles that contain neurotransmitter that are going to be activated so that neurotransmitter are going to be released. So more specifically, when a vesicle enters into this active zone, what we'll be interested in is the steps by which calcium enters through specific receptor, specific channel called calcium voltage channels. And it has been recognized that the co-localization between those calcium channels and the vesicle is very important for triggering this event of calcium release. Although it is known that after fusion, a vesicle can go back and be recycled, and when the vesicle are inside here, this reserve pool, it can move, although the exact mechanisms still remain to be understood, move into the active zone to do their function. So if we look specifically again at this region and look at the region between the vesicle and the membrane, we see that the calcium voltage channel, when calcium enter, first of all, this calcium voltage channel could interact with a specific protein that are associated with the vesicle, and calcium has to bind a fundamental protein, the syntaxin, so that calcium can trigger the snare complex, so that the vesicle here will fuse. So our calcium ion find their target underneath the vesicle has remained an important question, because when calcium goes in, usually they move by Brownian motion. Maybe there is some effect here of the electrical field, although it's not clear. But the calcium ion can either be lost here, or some of them can bind to this syntaxin molecule. And there are a few sites. So calcium needs to be bound to 4 here represented as two, but to several sites in order for the snare to be activated. So how this probability to bind depends on the calcium voltage organization and the exact local geometry, because it's clear that underneath a vesicle you have here the geometry of a ball, which is just nearby the plasma membrane. So about 10 years ago, the concept was the following. Calcium goes in, it creates a local domains, which was called calcium nanodomains, which can persist, let's say, for a few milliseconds. And depending on the distance between these calcium channels and the vesicle, there has been several predictions. So this is the distance, this is the calcium concentration in this area, and this prediction were made basically when there was no 3D geometry. So then several hypotheses can be explored. Either the calcium voltage channel are uniformly distributed here, the red spots, in a field of vesicles. Here they could be aggregated, they could be partially aggregated. And probably this the different configuration could happen and lead to different type of release. For example, when the calcium are far from a vesicle, probably a large amount of calcium is needed to trigger the close proximity located vesicles. While if we have here a cluster, maybe and the distance is smaller, then of course you need less calcium voltage channels to get this probability of having this few calcium bound. So this was some kind of results where the log of the release probability as the function of the flux could be fitted with some exponents, such as here this is a slope 1 and slope 3.5, and at that time this indicated some kind of cooperativity between those calcium sensors. So 3.5 means you need several cooperative binding sites for activation. So we wanted to challenge this vision in collaboration with my collaborator Martin Einer, and for this what he developed a way to follow the different calcium voltage channels, and for this he made construction where he added a small fluorophore on these calcium voltage channels and look at 3 different types depending of the length here of the C-terminus. So there was basically the short and long type calcium voltage 2.1 channels. And so the idea is, what are they really doing when we look at them under the microscope? And so this was the result. First of all, here is an example of an axon where you see in white calcium activation. So here calcium is activated. This is the electrophysiological responses as a function of time in 1, 2, 3, and 4 and 5 different regions. And what can be seen is that you see the different colors here, those corresponds to the trajectories, and the trajectory are actually accumulated in this specific region where calcium can be seen, and this is associated with a marker of the synapse, which is called this synaptophysin marker. And in addition, these calcium voltage channels are forming some type of nanodomains where they can stay longer, they aggregate, forming here a region where you can see these arrows of different color converging, and this can be translated into an energy, such as here 2.1 or 3.5 kT. And this suggests that there are specific mechanisms to trap transiently those calcium voltage channels. So this is a much better resolution a figure where you see here these different trajectories. So each trajectory corresponds to the path of a single calcium voltage channels. And here when you see the magnification, we have emphasized here through this ellipse this region of high density where trajectory aggregates. So if you look at the density, you see a high density at the center, and we can calculate at each point the diffusion coefficient at the membrane as described or seen by these calcium voltage channels. And here is an example of a vector field which gives us the information about the fact that they are trapped. You see these arrows are converging toward this region. And here this is another example where we have the trajectory, the density, the diffusion coefficient, and the associated vector field. So this shows that these calcium voltage channels are not fixed, but rather moving inside this presynaptic terminal. Another interesting part of this analysis is the fact that this region where the calcium voltage channel are trapped, if we look at this region over time, that is after 10 seconds, 20 seconds, 30 seconds, we see that the region of high density can deform, and sometimes it can appear just transiently. And so when we looked at the analysis, when this boundary, which is something we calculated, disappears, we define this as the end of the presence of these potential wells. So we could then, as a function of time, look at how the energy varies, calculated from a method that we developed with my student Pierre Pareto. And when we estimated the time where the nanodomains persisted, we found that for these three different cases of calcium voltage channel 2.2, 2.1 with long and short tail, we found about 30 seconds for the lifetime of this potential well, which give us an indication of the stability of this region of high density. So now this is an example we have here on an axon, and we look through this potential well, this region of high density, how they are distributed. And as you can see here, they are distributed at different places. And for each places, we can estimate here the energy of this well. So in this presentation, I show that the presynaptic calcium voltage channel, they are forming nanodomains. And inside these nanodomains, calcium can enter through this calcium voltage channel that are transiently trapped. We estimated in the past that this region were about of the size of 80 nanometer, which mean that they are probably encountering about one vesicle. So in the next YouTube, I'll explain how to compute the release probability by accounting for the distance between the calcium voltage channel, at least the distance between the potential well center and the center of the vesicle. So how this depends on the distance between the vesicle and the channel, and how can we estimate the probability through the time it takes for channels to be activated.